Good evening, everybody. Myself, S. Ramachandran, Associate Professor, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences, Vistas, Chennai, is going to present a seminar on the topic Amino acids and its properties. Introduction. Amino acids are the building blocks of the protein. There are 20 naturally occurring amino acids. All 20 amino acids have common structural features. All amino acids have an amino group, a carboxylate group, a hydrogen bonded to the same carbon atom. They differ from each other in the side chain called R group. R groups vary in structure, size, electrical charge, and influence the solubility of amino acids in water. This is the basic structure of the amino acid, where you can find the amino group, the carboxyl group, the hydrogen, and the RL group. So you have different types of amino acids. One you have the aliphatic amino acids, aromatic amino acids, saturated amino acids, etc. So you have classification of amino acids based on nutrition, nothing but essential amino acids and non-essential amino acids. You also have based on the R group, nothing but the non-polar aliphatic R group, polar uncharged R group, aromatic group, positively charged and negatively charged R group. So you can find out the different polar uncharged R group. Here you can find out the aromatic groups like the phenylalanine, tyrosine and tryptophan. Here you can find out the positively charged groups like your lysine, arginine and histidine. Here you can find out the negative charge R group like aspartate and glutamate. Here you can find out the different 20 amino acids with their structures. So coming on to the essential amino acids and the non-essential amino acids. And essential amino acids are supplied in daily diet. They are not synthesized in the body. But non-essential amino acids need not be supplied in the body. The short form of essential amino acid is MATT will fly. MATTVILLPHLY. So this is the basic structure of the amino acid. As I told you, it is based on the projection rule, Fisher's projection rule. You have the positive charge and the negative charge. The properties of amino acids are, except glycine, they are chiral in nature. Amino acids exist as D form and L form that are non-superimposable mirror images of one another. L form occur naturally in proteins. The properties of amino acids are they form internal salts called the zwitterions. Both the positive and the negative charge takes place in the same amino acid structure. So you have the non-ionic form and the zwitterion form. The right hand side you can find the nitrogen carries the positive charge and the O carries the negative charge. That is why it is called as an zwitterion. So electronegativity property and the electropositivity property is being shown in the zwitterion. And when you want to find out the properties of an acid, it forms a peptide bond with two bonds. You have the monopeptide and the dipeptide, which gives the corresponding protein molecule. So you can see the peptide bond, CONH bond is called the peptide bond, and it is identified by the test called biuret, BIURET. Copper sulfate in sodium hydroxide gives a violet color, and it is being compared with the control with water. Therefore, if the purple charge comes, it means it is forming the presence of peptide bond. So what happens is the carboxyl group and the amino group reacts. Water molecule gets cleaved to form the peptide bond. So when you find out the amino acid residues, you have the two types. One is the C-terminal amino acid and the N-terminal amino acid, which means C-terminal means they have the group which is attached to the carbon, which is consists of the free carboxyl end, and the N-terminal consists of the free N group. So you can see over here, this is the carbonyl terminal end, this amino terminal end. So you have a free amino group and the free carbonyl group or carboxyl group. This is the peptide bond formed between alanine, glutamic acid, glycine, and lysine. Coming on to the amino acid metabolism, very important. It forms a peptide bond, therefore it forms the waste product by the formation of the ATP by combining ADP and pi, that is adenosine diphosphate and the phosphate group. And the role of amino acid, it is the protein monomeric unit and it acts as an energy source and the precursor of other biological molecules. The precursor of nitrogen containing compounds are the heme, nucleotide, amine, nucleotide coenzymes, and glutathione. Amino acids forms the pyruvate, the oxaloacetate, and the alpha ketoglutarate, which in turn forms the glucose and the fatty acids and the ketone body's metabolism. The first reaction in amino acid witnessing deamination, removal of amino group, is called the deamination, which undergoes the urea cycle, removal of the waste product. Next enzyme is transaminase or aminotransferase, where amino group is transferred from one compound to the another compound. You have another reaction called as oxidative deamination. Both oxidation and the deamination takes place together. Other one is amino acid oxidate, where oxidation takes place to form the keto alpha keto acid. So you can find out the formation of act of ketoglutarate by the oxidation amino acid oxidase enzyme, which is in turn goes to the nitrogen cycle. So the classic example of glutamate dehydrogenase, which is an oxidative deaminase reaction where glutamate forms the alpha ketoglutarate. The next one is formation of urea, removal of waste product, where aspartate reacts with carbon bicarbonate in the form and ammonium radical in the urea cycle to form urea. It is a cyclized product which takes place both in the liver, mitochondria, and in the cytoplasm. So removal of nitrogen waste. So nitrogen extraction patterns take place by aminotelic amino extracting units, ureotelic and uricotelic. So urea synthesis is nothing but called as urea cycle or Krebs-Henselt cycle. Krebs cycle is called as TCA cycle, but Krebs-Henselt cycle is called as urea cycle. It takes place in the 5 steps. 5 steps are involved in the urea synthesis, 2 steps in the mitochondria, 3 steps in the cytosol. It requires ammonium radical, carbon dioxide, ATP, and the aspartate. Therefore, in the liver, in the cytosol, in the mitochondria, many processes take place, where the first step is synthesis of carbamoyl phosphate, second step is the formation of citrulline, third step the formation of adenosine, fourth step the cleavage of adenosine, and the fifth step is formation of urea by cleavage of arginine. This is the classic representation of reaction taking place in the urea cycle with various structures which happens in the liver, mitochondria, and the cytosol. This is the overall urea cycle. Aspartate forms urea and fumarate, the last part of the cycle. And the regulation of urea cycle is done by allosteric, nothing but by the NAC synthesis, nothing but N-acetyl glutamate is your precursor or the regulation of a urea cycle. Now, amino acid can get degraded to many compounds present in the cycle, from the citric acid cycle to the glycol cycle. Therefore, you can find out what all amino acid forms what all compound. Like example, alanine, cysteine, glycine forms pyruvate acid, isoleucine forms acetyl-CoA, leucine-lysine forms acetoacetate, your arginine, glutamate, glutathione, histidine forms the aspartate. Your isoleucine forms the succinyl-CoA, the aspartate forms the fumarate, and the again asparagine forms the oxaloacetate. This shows that various amino acids get cleaved to corresponding compound, which is in turn used in the carbohydrates metabolism. So alpha ketoglutarate is generated from 5 amino acids: proline, glutamate, glutamine, arginine, and histidine. This is the basic reaction. Next comes to the ketone body. The 6 amino acids that are degraded to acetoacetyl-CoA and aceto or acetyl-CoA can be converted to the acetoacetate and beta hydroxyglutaryl-CoA, which is nothing but the formation of the ketone bodies in the urine. These are the references which I use for my seminar. Thank you.